This is Chapter 5, the Integumentary System. Now, the integumentary system is comprised of skin and its accessory structures, such as hair, nails, sweat glands, and sebaceous or oil glands. There are two distinct regions of the skin: an outer superficial layer called the epidermis, and an inner layer called the dermis. The dermis is located deep to the epidermis. Now, the epidermis consists of the specific tissue subtype, keratinized, stratified, squamous epithelial tissue. It is also avascular and innervated. Therefore, the epidermis lacks its own direct blood supply, but does contain nerve endings. The principal tissue type in the dermis is connective tissue, which is highly vascularized and innervated. The hypodermis is also alternatively known as superficial fascia or the subcutaneous layer. Note that the hypodermis is not a true layer of the skin because it lies directly beneath the dermis, which already constitutes the deepest layer of the skin. Therefore, the hypodermis is located deep to the skin. Here, both prefixes hypo and sub translate to deep or underneath. The hypodermis is mainly comprised of loose adipose connective tissue, which insulates against heat loss and absorbs shock. We rely on the hypodermis to anchor skin to underlying muscle. Now, the epidermis is comprised of either 4 or 5 layers, depending on the thickness of the skin region. Thick skin found in high abrasion areas, such as the hands and feet, contain 5 layers or strata. These 5 layers of the epidermis, from superficial to deep, are as follows: stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, and stratum basale. There are many mnemonics to help remember the layers of the epidermis. For instance, one helpful mnemonic is "come, let's get sunburned." Here, the C in come is for corneum, the L in let's is for lucidum, the G in get is for granulosum, the S in sun is for spinosum, and the B in burned is for basale. Now, thin skin, which covers most of the body, except for the hands and feet, only contains 4 layers or strata. Thin skin does not have a stratum lucidum layer. Therefore, the stratum lucidum is only found in thick skin. Now, the thickness of the stratum corneum is the primary factor that determines whether a skin region is deemed thick or thin. If the stratum corneum is thick, then the skin region is deemed thick as well. If the stratum corneum is thin, then the skin region is deemed thin. For example, when we compare the two photomicrographs below, we can see that the image on the right has many more layers of dead, keratinized, and flattened cells in the stratum corneum, compared to the image on the left, whose stratum corneum is much thinner, with fewer layers of dead keratinized cells. Here we have a reference photo of the skin at 40x total magnification, which provides an overview of the two major skin layers, the epidermis and dermis, as well as the underlying hypodermis. Out of the two major skin layers, we can see that the thicker layer is indeed the dermis. The dermis makes up a large portion of the skin's overall thickness. Now, the deepest or base layer of the epidermis is the stratum basale, which is adjacent to the basement membrane that adheres the epidermis to the underlying dermis. The stratum basale consists of a single row of stem cells that are mitotically active, continually dividing and producing two daughter cells from each original stem cell. Recall that a stem cell is a non-terminally differentiated cell that can self-renew and differentiate into specialized cell types. Here, the fate of each daughter cell is as follows. One daughter cell differentiates into a specialized cell called a keratinocyte on its journey from the basal layer all the way to the free apical surface. Now, it takes approximately 1 month or between 25 to 45 days for this daughter cell to reach the surface. The other daughter cell remains in the stratum basale as a stem cell and essentially takes over as the new stem cell. Anywhere between 10 to 25% of the cells in the stratum basale are melanocytes, which appear as clear cells shown here. And there may also be tactile or Merkel cells, as well as dendritic cells present in the stratum basale as well. Above the stratum basale is the stratum spinosum, which is several cell layers thick and often described as the prickly layer. Numerous cell-to-cell junctions called desmosomes connect these keratinocytes tightly together, giving the cells a spiky or prickly appearance under the microscope. Here, the term desmo translates to bridge, so desmosomes are intercellular bridges. Keratinocytes in this layer contain a web-like system of pre-keratin filaments which attach themselves to desmosomes. These pre-keratin filaments allow the keratinocytes to resist tension and pulling. Now, scattered among the keratinocytes in the stratum spinosum layer are abundant immune cells called dendritic cells. Now, immediately above the stratum spinosum is the stratum granulosum, a thin layer of living keratinocytes named after their grainy or granular appearance due to the abundance of dark staining granules present. Now, the process of keratinization officially begins in the stratum granulosum. This is where keratinocytes produce keratin. Keratinocytes in the stratum granulosum will exhibit a drastic change in their appearance, where the cells flatten and their nuclei and organelles disintegrate. These keratinocytes also accumulate two types of granules, keratohyalin granules and lamellar granules. Now, keratohyalin granules produce proteins that are secreted into the more superficial layers to help cross-link keratin filaments together. This creates a tight, impermeable barrier that protects the epidermis from invasion by foreign particles, as well as water loss. Lamellar granules secrete a glycolipid that coats the surfaces of keratinocytes, effectively creating a hydrophobic water barrier against water loss. Note that all cells above the stratum granulosum are too far from dermal blood capillaries and therefore too far from nutrient supply to survive. As a result, all cells in the layers above the stratum granulosum will die off. Directly overlying the stratum granulosum is the stratum lucidum. Here, the term lucidum translates to clear or translucent. Recall that the stratum lucidum is exclusively located in thin skin. It consists of a band of 2 to 3 rows of clear, flattened, and dead keratinocytes. Lastly, we arrive at the stratum corneum, which is the outermost layer of the epidermis consisting of flattened, dead, and anucleated keratinocytes. It is also called the horned layer because its cells are toughened like the horns of an animal as a result of increased keratinization. The keratinocytes in the stratum corneum slough off and are constantly being shed and replaced by the division of cells in the deeper layers. In thick skin, we expect about 20 to 30 rows of keratinized dead cells. Now, the stratum corneum accounts for approximately 3/4 of the epidermis's total thickness. The dead cells of the stratum corneum protect the deeper cells from abrasion and water loss. And these cells behave as a biological, chemical, and physical barrier. Now, there are 4 cell types found in the epidermis: keratinocytes, melanocytes, dendritic or Langerhans cells, and tactile or Merkel cells. The first cell type is keratinocytes, the main cells found in the epidermis. Keratinocytes produce keratin, a protein that affords skin its protective properties. Now, keratinocytes are tightly connected by a type of cell-to-cell junction called desmosomes. Many keratinocytes will slough off the surface of the skin each day, and we can expect to find keratinocytes in all epidermal layers or strata. The second cell type is melanocytes, which produce a pigment known as melanin. Melanin is packaged into organelles called melanosomes and then transferred to keratinocytes. Melanin shields and protects the nuclei of those keratinocytes from harmful UV radiation. Melanocytes are located in the stratum basale. The third cell type is dendritic or Langerhans cells. They are star-shaped, phagocytic white blood cells that patrol the two deepest layers of the epidermis. That is the stratum spinosum and the stratum basale. As key activators of the immune system, dendritic cells engulf foreign invaders and present them to other white blood cells, thereby alarming our immune system and activating our immune response. Lastly, the fourth cell type is tactile or Merkel cells, which are sensory receptors that detect touch sensations. Tactile or Merkel cells can be found in the stratum basale. Here we have a reference image of thin skin where the dead keratinized stratum corneum appears as lines flaking off. We will assume all purple epidermal cells that still contain their nuclei are living keratinocytes. We will also assume that any clear cell in the stratum basale is a melanocyte. Lastly, we will assume that the isolated purple cells in the dermis are fibroblasts. Now, in the photomicrograph on the left, we have pigmented skin, which is characterized by the presence of melanin in its stratum basale. Essentially, the more melanin present in the stratum basale, the more pigmented or darker in coloration the skin appears. In the photomicrograph to the right, we have non-pigmented skin, which is characterized by an absence of melanin in its stratum basale. Now, the less melanin present in the stratum basale, the less pigmented and thus the lighter in coloration the skin appears. Note that although individuals generally have the same number of melanocytes, it is the amount of melanin produced by their melanocytes that varies from person to person. Now, cell types found in the dermis include fibroblasts, macrophages, and occasionally white blood cells. In addition to nerves, blood vessels, and lymphatic vessels, the dermis also contains embedded extensions of epidermal structures, such as hair follicles, oil glands, and sweat glands. The dermis consists of two layers, the superficial papillary layer and the deep reticular layer. Now, the papillary layer comprises only about 20% of the dermis's overall thickness, and the reticular layer comprises the remaining 80% of dermal thickness. So the reticular layer is much thicker compared to the papillary layer. Now, the papillary layer of the dermis is composed of loose areolar connective tissue. This loose arrangement of collagen and elastic fibers allows phagocytic cells to patrol for invading microorganisms. In the papillary layer, we can see finger-like projections extending up into the epidermis. These finger-like projections are termed dermal papillae, and they are the basis for our fingerprint patterns. In addition to free nerve endings, dermal papillae also contain receptors called tactile or Meissner's corpuscles that detect light or gentle touch. Now, in contrast to the papillary layer, the reticular layer of the dermis is composed of dense, irregular connective tissue. The reticular layer is filled with a network of tough collagen and stretchable elastic fibers. Between the dermis and underlying hypodermis, there is a network of blood vessels called the cutaneous plexus. Now, because the reticular layer borders the hypodermis, some parts of the reticular layer's extracellular matrix may also contain pockets of adipose cells. While there is no distinct boundary between the papillary dermis and the reticular dermis, we are still able to see the loose and airy arrangement of loose areolar connective tissue in the papillary dermis transition into a compact arrangement of thick collagen bundles. This compact arrangement characterizes dense irregular connective tissue and thus signals that we are now in the reticular dermis. Here, this figure provides a schematic overview of the integumentary system, displaying the structures associated with each major skin layer. There are three pigments that contribute to skin color: melanin, keratin, and hemoglobin. Now, melanin is the only pigment that is produced in the skin. Melanin is synthesized from the amino acid tyrosine by the enzyme tyrosinase. And melanin is ultimately packaged into melanosomes that are sent to keratinocytes to protect their DNA from UV damage. The more sun exposure an individual receives, the more melanin will be produced. Note that there are two forms of melanin: reddish yellow and brownish black. The amount and type of melanin is the primary factor that determines skin color differences. When melanin accumulates locally on the skin, This manifests as freckles or pigmented moles. The next pigment is keratin, which is a yellow to orange pigment. Keratin is most pronounced in the stratum corneum of the palms of the hands and the soles of the feet. It also accumulates in the hypodermis as well. Keratin can be converted to vitamin A, which is essential for vision and epidermal health. The last pigment that contributes to skin color is hemoglobin, which is the red pigment found in red blood cells. Hemoglobin produces the pinkish hue that is seen in fair or light skin. Because individuals with fair or light skin produce lower levels of melanin, the color of hemoglobin shows through. Now, the scientific term for sweat gland is sudoriferous gland, which comes from the Latin word sudor, meaning sweat. With the exception of the nipples and various parts of external genitalia, all skin surfaces contain sweat glands. There are two main types of sweat glands, eccrine and apocrine sweat glands. Note that sweat glands contain modified muscle cells called myoepithelial cells that contract upon nervous system stimulation to drive sweat into a duct. Here, the prefix myo translates to muscle. Eccrine sweat glands are alternatively known as merocrine sweat glands. Here, the term merocrine is used to describe the mode of secretion whereby a cell secretes their product through exocytosis. And the secreting cell remains unharmed and intact during the secretion process. Eccrine sweat glands are the most widespread sweat gland in the body, and they are present in highest numbers in the palms, soles, and forehead. A helpful mnemonic is to remember the E in eccrine as E for everywhere in the skin. Ducts of eccrine glands connect to pores that will empty sweat onto the surface of the skin. Now, a core function of sweat glands is to control body temperature, otherwise known as thermoregulation. For this reason, stimuli for eccrine sweat glands includes elevated body temperature or activation by the sympathetic nervous system, our fight or flight response system. Eccrine sweat secretions consist of predominantly water, but also contains components such as salts, vitamin C, and metabolic wastes. Additionally, eccrine sweat also contains immune or defense components in the form of antimicrobial proteins called defensins, which kill bacteria. In the photomicrograph on the right, we can see a cross-section of eccrine sweat glands. The duct segment of eccrine glands are lined with stratified cuboidal epithelium, while its secretory portion is composed of simple cuboidal epithelium. Now, apocrine sweat glands are strictly localized to the axillary and anal genital regions. A helpful mnemonic is to remember the A in apocrine as A for axillary and anal genital. In the apocrine mode of secretion, apocrine cells release their product by shedding a part of the cell. The top portion of the cell, with the secretory vesicles that contain the product, is pinched off. Essentially, the portion of the cell that pinches off is considered the secretion itself. Now, unlike the watery and dilute sweat secretions of eccrine glands, apocrine gland sweat secretions are a viscous, milky, or yellowish sweat that contains fat and proteins. Now, the breakdown of apocrine sweat by bacteria will lead to body odor, since ammonia is released as a byproduct of that metabolism. Apocrine glands also contain ducts that empty into hair follicles instead. The function of apocrine sweat glands in humans is unknown, but in certain mammals it is speculated that apocrine glands may behave as pheromones or sexual scent glands to attract opposite mates. Examples of modified apocrine glands include the mammary glands of the breast, which secrete milk product. Now, our next accessory structure is the hair follicle, which is a tube-like sheath that functions in the production and nourishment of hair and is essential for the maintenance of hair growth. Since hair follicles develop from keratinocytes of the epidermis, this means hair follicles will be largely composed of epithelial tissue with a smaller proportion of connective tissue. The portion of the hair that is visible will be located above the skin, and this is termed the hair shaft. Now, the portion of the hair that is not visible will be located below the skin, and this is termed the hair root. Hair follicles only surround the hair root, but not the hair shaft. Here we see several longitudinal sections of the hair follicle surrounding the hair root. We also see a cross-section of the hair root in the center, with its entire circumference surrounded by a hair follicle. Now, the erector pili is specialized smooth muscle that attaches to each hair follicle. When it contracts, the erector pili muscle causes the hair to stand erect and a goosebump forms on the skin. This brings us to sebaceous glands, the scientific term for oil glands. Sebaceous glands are widely distributed throughout the skin, except for the palms and soles. The majority of sebaceous glands develop from hair follicles and secrete their oil or sebum product directly into hair follicles. This means sebaceous glands will share a similar tissue makeup to hair follicles. As such, sebaceous glands are composed of epithelial tissue as well. Now, sebaceous glands remain quite inactive until puberty. So the primary stimulus for sebaceous gland activation is hormones, particularly a class of masculinizing steroid hormones called androgens. Their secretion is oil or sebum, which contains a mixture of lipids. Sebaceous glands undergo a holocrine mode of secretion, where the secreting cell releases its secretions by rupturing its cell membrane, which ultimately destroys the cell. Because the secreting cell dies to become the secretory product itself, this process is considered the most damaging type of secretion. In terms of sebum's protective function, sebum has bactericidal or bacteria-killing properties and is the first line of defense against infection. Moreover, sebum also has moisturizing effects and helps to keep hair and skin soft and pliable. While the skin's primary function is to serve as a barrier for protection, it also has many other bodily functions, including body temperature regulation, cutaneous sensations, metabolic functions, serving as a blood reservoir, and waste secretion. Now, the skin acts as a protective chemical barrier by secreting various chemicals such as sweat and sebum to defend against invading bacteria. The surface of our skin also contains a thin film called an acid mantle that lowers the pH environment of our skin in order to curb the growth and replication of microbes. Lastly, melanin also serves as a chemical barrier by protecting the nuclei of keratinocytes from UV radiation damage. Now, the dead keratinized cells of the stratum corneum allow our skin to behave as a physical barrier to water and water-soluble substances. However, there are some chemicals that still have limited penetration of the skin, and this includes lipid-soluble substances, plant oleoresins such as poison ivy, medications such as nitroglycerin, and certain drug agents that help carry other drugs across the skin. Additionally, cells present in the epidermis and dermis allow the skin to act as a biological barrier. In the stratum spinosum and stratum basale of the epidermis, phagocytic cells, known as dendritic cells, engulf any invading substances, such as pathogens, that they deem foreign. These foreign substances are known as antigens, which are ultimately presented to other white blood cells to activate an immune response. The dermis contains another type of phagocytic white blood cell called macrophages that also activate the immune system by presenting foreign antigens to other white blood cells. Now, the skin plays a critical role in the body's thermoregulation system. When there is a rise or elevation in body temperature, blood vessels in the dermis will dilate or widen, and sweat gland activity also increases to cool the body back down to homeostatic range. Here, vasodilation is the process by which blood vessels expand or widen and move closer to the skin's surface, resulting in increased blood flow toward the skin surface. Increased blood flow beneath the epidermis allows heat to escape from the skin surface, resulting in increased heat loss. With respect to sweat evaporation, As sweat evaporates from the skin, heat is exchanged with the environment and ultimately lost from the body, which contributes to a decrease in skin temperature as well. Now, when there is a fall or drop in body temperature, blood vessels in the dermis will constrict or narrow to slow the loss of heat. Here, vasoconstriction is the process by which blood vessels narrow or constrict to pull away from the skin's surface, resulting in decreased blood flow towards the skin surface. Decreased blood flow beneath the epidermis causes less heat to escape from the skin surface, resulting in decreased heat loss. Now, cutaneous sensory receptors called exteroreceptors are located in the skin and are responsible for detecting external stimuli such as temperature and touch. This allows the nervous system to respond to changes in the surrounding environment. Additionally, sensory receptors, known as free nerve endings, are primarily responsible for relaying information to the nervous system when the body is exposed to painful or damaging stimuli. In terms of metabolic functions, the skin is able to synthesize vitamin D, required for the intestinal absorption of calcium. Moreover, skin also produces an enzyme called collagenase that enhances the natural turnover of collagen to prevent wrinkle formation. Now, skin also serves as a blood reservoir, holding up to 5% of the body's entire blood volume. In response to certain stimuli, blood vessels in the skin can also be constricted, such that blood is redirected towards other organs, such as an exercising muscle or the heart. Lastly, the skin functions in the excretion of waste products. The skin can secrete limited amounts of nitrogenous waste, such as ammonia, urea, and uric acid. And sweating can also result in the excretion of water and salt.